And I'd like to thank the organizers for inviting me to give this presentation. In measuring persistent HIV infection, there are several factors that we should take into account. In which body compartments should we measure persistent HIV infection? Should this be peripheral blood, gut-associated lymphoid tissue or other lymphoid tissues, or brain? And does measurements of persistent HIV in the blood really represent what takes place in the tissue? And I think Tim Shacker, our next speaker, will give us some thoughts and educate us about that. But also, in which cells should we measure persistent HIV? Should we measure persistent HIV in resting memory CD4 T cell subsets, naive T cells, memory stem cells, or cells from the monocyte and macrophage lineage? What status of the virus? What is the status of the virus? Is it replication competent? Are we measuring replication competent virus? Are we measuring replication incompetent virus? And finally, what is the status of infected cells? Are they quiescent or activated? We have several current methods that we use for evaluating curative strategies. And all of these methods have advantages and disadvantages. The first is the measurement of plasma-derived HIV RNA, usually using the single copy assay or other variants of this assay. This is a relatively inexpensive assay, but it does not, although it can measure virus that's released into the plasma when latently infected cells are reactivated, this assay does not directly detect the frequency of latently infected cells. And furthermore, patients on long-term ART have a measurement of plasma RNA that is very close to the limit of the assay detection. The quantitative viral outgrowth assay is a direct measurement of replication-competent virus, or the number of proviruses that are capable of causing a productive infection. However, this assay requires large quantities of cells, is expensive, time-consuming, and does have a limited dynamic range. And this assay does not detect, as Bob Silicano explained and presented yesterday, it does not detect all proviruses which pose a barrier to a cure. There are also T-cell activation assays in which cells are activated and multi-spliced RNA transcripts are measured. And these assays do not require less culture time and no need for an outgrowth viral assay. But these assays may detect some defective viruses. And similarly to the viral outgrowth assay, these assays do not detect all proviruses which pose a barrier to a cure. The measurement of total HIV DNA, whether it's by qPCR or digital droplet PCR, is also, and this is total intracellular HIV DNA, is also inexpensive, easy, and quick. And for digital droplet PCR, it's an absolute quantification. But this assay, as was described by Catherine, does detect defective proviruses, which may not pose a barrier to a cure. Integrated HIV DNA, measured by ALU PCR, excludes unintegrated HIV DNA and has less error than the viral outgrowth assay, but this assay can detect defective proviruses and does not detect proviruses that are too far from the ALU sequence. And finally, we have cell-associated RNA, whether cell-associated intracellular RNA, total RNA measurement, unspliced RNA, or multi-spliced RNA. This assay is inexpensive, easy, quick, and does measure intracellular HIV RNA transcripts. However, this assay can detect aborted and defective HIV RNA transcripts, which may not be translated into viral proteins, and if they are translated into viral proteins, these viral proteins may not produce an infectious virion. So today I would like to share with you 3 new methods for evaluating curative strategies. And the first of these assays was developed to answer the question, can the host immune response against HIV be used to quantify the latent HIV reservoir? In other words, can antibodies directed against specific HIV antigens, such as the GP120, the RT, the P24, their titer, avidity, and specificities, can they be used to measure peripheral reservoir the peripheral reservoir or the tissue reservoir. To answer this question, an assay was developed by Peter Burbello at the NIH, and this is the Luciferase Immunoparticipation Assay, or LIPS. And this assay utilizes recombinant proteins that are chimeras of light-producing Rhinella luciferase and a pathogen-specific antigens, or HIV-specific antigens, such as GP120, GP41, reverse transcriptase. And in fact, these chimeras can essentially contain the whole HIV proteome. And these antigens are then added, and these antigens, these chimeras are then added to diluted patient samples. And then IgG beads are added, which bind the antigen-bound antibodies. These antigen-bound antibodies can then be washed, and then the amount of antibody directed towards a specific antibody can be quantified by a luciferase readout of light units. This assay has a very broad dynamic range, from 10 copies to 10 to the 7th copies. And the higher the light units readout, or the more light units that are read out or measured by this assay, is equated to a higher amount of antibody towards a specific HIV antigen. So this method was used to determine the antibody response against 8 HIV proteins for several participant groups, including uninfected persons, the Berlin patient, elite controllers, HIV-infected persons prior to and after antiretroviral therapy. And what was found was the loss of antibody response to P24 appears to be associated with the cured state. In other words, the antibody response found in the uninfected group of participants and the Berlin patient was much lower than the antibody response to the GP24 antigen than infected HIV individuals who were elite controllers or HIV-infected persons prior to or after antiretroviral therapy. However, across all proteins, the antibody response to the GP41 tended to show the most consistent group-to-group differences, with increasing responses across the uninfected, and then a higher response to the GP41 antigen by the Berlin patient, higher response by the elite controllers, and the highest response in HIV-infected individuals who are not on antiretroviral therapy, with a lower response of those HIV-infected individuals who are on antiretroviral therapy. Other measures of the HIV reservoir were found to correlate with this antibody assay, and most specifically, antibodies directed against the GP120, GP41, and the RT were found to be strongly correlated with total digital droplet PCR or integrated ELU PCR quantification of HIV RNA. Other antibodies directed against the GP41 and the GP120 were also found to have moderate correlations with other reservoir measures. The second assay I'd like to share with you today is the improved HIV quantification assay, which is developed by Hologic and is fully automated on the Panther system. This assay involves 3 steps. First, the target HIV RNA is captured by a bead-based extraction, and this is specific for HIV RNA. This RNA is then amplified by a transcription-mediated amplification process, and then this amplified RNA target is quantified using a real-time detection technology. So looking a little closer at the magnetic bead technology or extraction technology, 700 microliters of plasma are treated with a detergent to lyse cell-free virions. These virions, once lysed, release a RNA target, and the target RNA sequence shown in blue is then captured by a capture oligomer shown in gold, and this capture oligomer is also bound to a polyoligomer, which is bound to a magnetic particle. So by capturing this RNA target sequence, or this RNA by its target sequence, you can isolate the target RNA can be isolated, it can be washed and purified, and then released into a specific elution volume. Once the RNA target is captured, it is then amplified by transcription-mediated amplification, or TMA. This involves a Maloney murine leukemia virus RT, which contains a promoter for a sequence for T7 RNA polymerase, which converts RNA to double-stranded DNA. Two regions are amplified, the pol and LTR regions, using primer specific for these regions. Once the double-stranded DNA is produced, the T7 RNA polymerase produces multiple copies of an RNA amplicon from the DNA copy template. And this is done in sort of a rolling circle format in which multiple RNA amplicons are produced, making this, increasing the sensitivity in quantifying the target RNA region. Detection is achieved using single-stranded nucleic acid torches, which hybridize to the RNA amplicon and fluoresce. This assay has been extensively validated against other commercial assays for viral loads greater than 100 copies, as shown here. So the APTIMA Quant DX assay is highly correlated to other assays, but at viral loads greater than 100 copies per mil. So to evaluate the performance of this assay at lower copy numbers, it requires using larger volumes and assays that simply by running more replicates. So you can bring down the sensitivity of this assay by running multiple replicates of your plasma samples. And this is shown here. Blinded samples from 50 elite controllers who were subtype C were analyzed. 9 replicates were used to measure the viral RNA in the plasma of these elite controllers who had very low RNA levels by the APTIMA HIV quantitative TMA assay. And this was compared to 20 replicates which were used to measure these same samples using the HIV qualitative assay. And there was excellent correlation between these two assays with the number or percentage of replicates that were found to be positive in these low level, in these patients who have very low RNA levels. Also, all 20 of the negative control samples, or the control samples which were negative by all of the by all assays and all replicates. The third assay I would like to share with you is the murine viral outgrowth assay, the MVOA. And this assay, immunocompromised mice, which lack B cells, T cells, and NK cells, are engrafted with human T cells, and they develop a graft versus host disease. So in conducting this assay, 100 to 50 million PBMCs, or CD4 T cells, from HIV-infected individuals who have undetectable viral loads are injected into each mouse. And to further activate these cells in this humanized mouse model, anti-CD3 antibody can be injected into the mouse as well. And to deplete any CD8 T cells, which might actually have an antiviral effect, anti-CD8 antibodies can also be injected into the mice. And the viral outgrowth in these mice is measured by plasma viremia levels, which are measured by qPCR through weekly bleeds and in a terminal bleed at about 60 days. So using the MVOA, the sensitivity of the MVOA was then analyzed to see how well this assay could detect HIV from patients or participants on suppressive ART. So cells from participants who were on suppressive ART for 1 to 6 years and who had undetectable viral loads for the last year, 25 to 55 million PBMCs from these participants and 4 different participants were injected into 4 different mice, so 1 mouse per participant. On day 7, anti-CD8 antibodies were injected into the mice, and that's shown by the black arrows. The mice were bled weekly to evaluate the viral loads, and CD4 T cell counts were also quantified. And the percent of human origin CD4 cells are shown in blue on these graphs, But what's really exciting is that the MVOA detected replication component virus in all of these patients, as shown by the closed red circles. So all 4 patients, this assay was able to detect replication component virus. To further test the sensitivity of this assay, cells from elite controllers with undetectable viral loads were injected into the mice, either CD4 T cells or PBMCs. On day 7, and these mice were exposed to anti-CD8 antibodies, as shown by the black arrows, and sometimes multiple times. They were also exposed to anti-CD3 antibodies, as shown by the yellow arrows, some of the mice. The mice were bled weekly to evaluate the viral load and the CD4 T cell count. And again, shown in blue is the percent of human origin CD4 T cells in the graphs. And as shown for the patients on suppressive therapy, the MVOA was able to detect replication component virus from all of the elite controllers analyzed, as shown by the filled red circles. And finally, the question is, how many cells and how many mice can you use for one patient? This was done in collaboration with Hiro Johantino and Stephen Deeks at UCSF, and with Kelly Metcalf-Pate and Joel Blankson, who actually developed this assay and shared these slides with me. And this is a patient who was a lead controller on suppressive ART. And this participant had a leukapheresis, and 350 million CD4 T cells were purified from this participant's blood sample. 50 million cells were injected into 7 individual mice. So 350 million cells, 50 million cells into 7 mice. These mice were given anti-CD8 and anti-CD3 antibodies. But what was really exciting is that 3 out of 7 mice showed replication, or there was replication virus was quantified in 3 out of the 7 mice. So this shows that you can use a lot of cells from one patient who has a very low RNA level to see if this assay can actually show, this assay can be used for very high cell numbers from one patient with a very low RNA level to determine whether there is replication component virus still present in their cells. And in this case, there was. And 3 out of 7 mice had replication component virus grow out from the cells of this participant. So in conclusion, new assays are being developed for measuring the HIV reservoir. However, additional assays are still needed to differentiate between defective and replication component virus. One such assay could be a high-throughput, full-length proviral sequencing, and this was described by Bob Silicano yesterday. And full-length sequencing can identify defective virus. But the question still remains, must all potential reservoirs be analyzed? If not, are we confident that certain reservoirs are determinative of a cure or a remission? And finally, looking ahead, to determine the effectiveness of curative strategies, our field will need to develop a more standardized assay system which is sensitive, efficient, less costly, and adoptable in local settings, especially in resource-limited settings. I would like to thank my lab who put up with me for flying all over the world and giving presentations. I would like to thank the researchers at Hologic who were very helpful in the preparation of this presentation. I would like to thank Michael Bush, Doug Richman, Marta Luna, Kelly Metcalf-Pate, and Joel Blankson for sharing their slides with me and discussing how these assays do function. Our funders, but most particularly I'd like to thank with gratitude the study participants. Without their participation in these studies, we would not be able to develop new and more sensitive assays. Thank you.